Continuation of the chapter about eye. So, iris, conjunctiva, cornea, lens, anterior chamber, ciliary body, ciliary muscle, vitreous humor, fovea centralis, retina, choroid, sclera, these are the major regions. So, the lens is bathed on one side by aqueous humor and supported on the other side by vitreous humor. It has no blood supply but is metabolically active. It obtains nutrients from the aqueous humor. The lens is mostly water and proteins. The majority of vertebrate lens proteins are the alpha, beta, and gamma crystallins. Anatomically, the wall of the eyeball consists of three layers: fibrous tunic, vascular tunic, and retina. The fibrous tunic is the superficial coat of the eyeball and consists of the anterior cornea and posterior sclera. The cornea is transparent coat that covers the colored iris. Because it is curved, the cornea helps focus light onto the retina. Its outer surface consists of non-keratinized stratified squamous epithelium. The middle coat of the cornea consists of collagen fibers and fibroblasts, and the inner surface is simple squamous epithelium. The sclera, the white of the eye, is a layer of dense connective tissue made up mostly of collagen fibers and fibroblasts. The sclera covers the entire eyeball except the cornea. The vascular tunic or uvea is a middle layer of the eyeball. It is composed of three parts: choroid, ciliary body, and iris. Choroid is highly vascularized structure, which is the posterior portion of the vascular tunic. It is deeply pigmented with melanin. It provides blood supply and absorbs scattered light. The choroid coat forms the iris in the front of the eye. In the anterior portion of the vascular tunic, the choroid becomes the ciliary body. Ciliary body is located behind the iris and produces aqueous fluid that fills the front part of the eye. It maintains the eye pressure and also allows focusing of the lens. Iris present inside the anterior chamber. It acts like a diaphragm of the camera, dilating and constricting the pupil to allow more or less light into the eye. The retina is a light sensitive inner layer of the eye that covers about 65% of its interior surface. It contains the light receptors, the rods and cones. The photosensitive cells in the retina convert incident light energy into signals that are carried to the brain by the optic nerve. All the nerve impulses generated in the retina travel back to the brain by way of axons and optic nerve. At the point on the retina where the approximately 1 million axons converge on the optic nerve, there are no rods or cones. This point is called the blind spot, as this is insensitive to light. Rods and cones are both photosensitive but respond differently to different frequencies of light. They contain different pigmented photoreceptor proteins. Rod cells contain the protein rhodopsin, and cone cells contain different proteins for each color change. Rhodopsin is a transmembrane protein of rods, breaks down into opsin and retinal. Iodopsin of cones breaks down into photopsin and retinal. Rod vision. Rhodopsin is the light absorbing pigment of the rods. This G protein coupled receptor or GPCR consists of an opsin of 348 amino acids coupled to retinal. This physical change in retinal triggers the following chain of events, culminating in a change in the pattern of impulses sent back along the optic nerve. The formation of all transretinal activates its opsin. Activated rhodopsin in turn activates many molecules of G protein called transducin. So formation of all transretinal activates its opsin. Activated rhodopsin in turn activates many molecules of G protein called transducin. Transducin activates an enzyme that breaks down cyclic GMP. The drop in concentration of cGMP closes Na plus channels in the plasma membrane of the rod. Because these positive ions can no longer enter, the interior of the cell become more negative, hyperpolarized. This hyperpolarization of the cell leads to decreased release of transmitter molecules, glutamate at the synapse. The reduction in glutamate release activates some interneuron pathways, inhibits others. The interplay of excited and inhibited interneurons modulate the spontaneous firing of the ganglion cells to which they are connected. So once again just read this. Formation of all transretinal activities, its opsin activated rhodopsin in turn activates many molecules of G protein called transducin. Transducin activates an enzyme that breaks down cyclic GMP. The drop in concentration of cGMP closes Na plus channels in the plasma membrane of the rod. Because these positive ions can no longer enter, the interior of the cell becomes more negative or hyperpolarized. This hyperpolarization of the cell leads to decreased release of transmitter molecules, glutamate at the synapse. This reduction in glutamate releases activates some interneuron pathways, inhibits others. This interplay of excited and inhibitor interneurons modulates the spontaneous firing of the ganglionic cells to which they are connected. So cone vision, although cones operate only in relatively bright light, they provide us with our sharpest image and enable us to see colors. Two or three types of cone photoreceptors and a single type of rod photoreceptor are present in the normal mammalian retina. Some non-mammalian retinas have even more cone types. Most of the 3 million cones in each retina are confined to a small region just opposite the lens called the fovea or fovea centralis. It is the center of eye's sharpest vision and the location of most color perception. The 3 types of cones provide us with the basis of color vision. Cones are turned to different portions of the visible spectrum. Red absorbing cones, those that absorb best at the relatively long wavelengths peaking at 565 nm. Greens absorbing cones with a peak absorption at 535 nm. Blue absorbing cones with a peak absorption at 440 nm. Retinal is the prosthetic group for each pigment. Differences in the amino acid sequences of their opsins account for the differences in absorption. Like rods, the absorption of light does not trigger action potentials but modulates the membrane potential of the cones. So, rods used for scleropic vision, very light sensitive, loss causes night blindness, not present in fovea, slow response to light, one type of photosensitive pigment, confer achromatic vision. But cones used for phototopic vision, not very light sensitive, less loss causes legal blindness. Concentrated in fovea, faster response to lights, 3 types of photosensitive pigment in humans, confer color vision. Now let's learn about kidney. Renin, also known as angiotensinogase, is a circulating enzyme released mainly by juxtaglomerular cells in the juxtaglomerular apparatus. JGA of the kidneys in response to low blood volume or low body NaCl content mediated through the release of prostaglandins. Renin activates the renin-angiotensin system by cleaving angiotensinogen produced in the liver to yield angiotensin 1, which is further converted into angiotensin 2 by specialized cells of the lung capillaries. Angiotensin 2 then constricts blood vessels, increases the secretion of ADH and aldosterone, and stimulates the hypothalamus to activate the thirst reflex, leading to increased blood pressure. So, as a part of endocrine system, let's learn some hormones of gastrointestinal hormones. Gastrointestinal hormones are proteinaceous in nature. Some important gastrointestinal hormones and their roles are given below. Gastrin, G cells of pyloric glands and duodenum, stimulates gastric juice, gastric mucosa growth and pepsin secretion, and stimulate pancreatic secretion. Then secretin, duodenal and jejunum mucosa, stimulate pancreas, possibly liver, to secrete fluid base of pancreatic juice. It inhibits stomach movement and secretion of gastric juice. Then pancreozymin, duodenal and jejunum mucosa, stimulates pancreas to secrete enzymes. Then cholecystokinin, small intestine, stimulates the gallbladder for rhythmic contraction. This results in an increase in the rate of bile secretion. Then enterocrinin, intestinal mucosa, stimulates the intestinal mucosa to secrete the intestinal juice. Gastrin, secretin, pancreozymin, cholecystokinin, enterocrinin. Now let's learn about respiratory systems. The respiratory system consists of the nose, pharynx or throat, larynx, voice box, trachea, windpipe, bronchi and lungs. The respiratory system has following functions: help in gaseous exchange, intake of O2, food delivery to body cells, and elimination of CO2. The respiratory system consists of the nose, pharynx, larynx, trachea, bronchi, and lungs. The respiratory system has following functions: help in gaseous exchange, intake of O2, food delivery to body cells, and elimination of CO2 produced by body cells, helps to regulate blood pH, contains receptors for the sense of smell, filters inspired air, produces vocal sounds, phonation, and excretes small amounts of water and heat. Then respiration, the process of gas exchange in the body called respiration, has three basic steps. Pulmonary ventilation, pulmon in the sense it's lung, or breathing is the inhalation, inflow, and exhalation, outflow of air between the atmosphere and the alveoli of the lungs. Air flows between the atmosphere and the alveoli of the lungs because of alternating pressure differences created by contraction and relaxation of respiratory muscles. This is about pulmonary ventilation. Pulmonary ventilation. So pulmonary ventilation or breathing is the inhalation, inflow, and exhalation, outflow of air between the atmosphere and the alveoli of the lungs. Air flows between the atmosphere and alveoli of the lungs because of alternating pressure differences created by contraction and relaxation of respiratory muscles. Then external pulmonary respiration is the exchange of gases between the alveoli of the lungs and the blood in pulmonary capillaries across the respiratory membrane. In this process, pulmonary capillary blood gains O2 and losses CO2. Then internal respiration or tissue respiration is the exchange of gases between blood in systemic capillaries and tissue cells. In this step, the blood loses O2 and gains CO2. Within cells, the metabolic reactions that consume O2 and give off CO2 during the production of ATP are termed cellular respiration. So pulmonary ventilation, after that external respiration, then internal respiration. Next topic is about mechanics of breathing. Humans possess a pair of lungs located in the chest or thoracic cavity. The two lungs hang free within the cavity, being connected to the rest of the body only at the one position where the lungs, blood vessels, and air tube enter. This air tube is called bronchus. It connects each lung to a long tube called trachea, which passes upward in the body, past the voice box or larynx, and opens into the rear of the mouth. Air normally enters through the nostrils, which are lined with hairs that filter out dust and other particles. As the air passes through the nasal cavity, extensive array of cilia on its epithelial lining further cleans the air and moistens it. The air then passes through the back of the mouth, crossing the path of food as it enters first the larynx and then the trachea. From there it passes down through the bronchus and the bronchioles and into the alveoli of the lungs. The human respiratory apparatus is simply in structure, functioning as a one-cycle pump. The thoracic cavity is bounded on its sides by ribs which are capable of flexing, and on the bottom by a thin layer of muscle, the diaphragm, which separates the thoracic cavity from the abdominal cavity. Each lung is covered by a very thin, smooth membrane called the pleural membrane. The second pleural membrane marks the interior boundary of the thoracic cavity, dividing it into two halves. Each lung is thus suspended in its own cavity. Within the cavity, the weight of the lung is supported by water, the intrapleural fluid. During inhalation, the walls of the chest cavity expand, the rib cage moves upward and outward, and the diaphragm moves downward by stretching. In effect, we have enlarged the outer balloon by pulling it in all directions. This expansion of the fluid space causes the fluid pressure to decrease to a level less than that of the internal air pressure within the lung. As a result, the wall of the lung is pushed out. As the lung expands, its internal air pressure decreases and air moves in from the atmosphere. During exhalation, the ribs and diaphragm return to their original resting position. In doing so, they exert pressure on the fluid. This pressure is transmitted uniformly by the fluid over the entire surface of the lungs, forcing air from the internal cavity back out to the atmosphere. Breathing the cycle of inhalation and exhalation. This depends on the regular contractions of the muscles surrounding the thoracic cavity. The rate of breathing is regulated by the respiratory center in the brainstem. So here we could see a picture which shows this atmospheric pressure 760 mmHg at rest, diaphragm relaxed, alveolar pressure 760 mmHg, intrapleural pressure 756 mmHg, then atmospheric pressure 760 mmHg, then during inhalation diaphragm contract, atmospheric pressure 760 mmHg, and alveolar pressure 760 mmHg, intrapleural pressure 756 mmHg. During exhalation, diaphragm relaxing. Pressure changes in pulmonary ventilation. During inhalation, the diaphragm contracts, the chest expands, the lungs are pulled outward, and alveolar pressure decreases. During exhalation, the diaphragm relaxes, the lungs recoil together to inward, and alveolar pressure increases, forcing air out of the lungs. Then exchange of oxygen and carbon dioxide. The exchange of oxygen and carbon dioxide between the alveolar air and pulmonary blood occurs via passive diffusion, which is governed by the behavior of gases as described in two gas laws, Dalton's law and Henry's law. According to Dalton's law, each gas in a mixture of gases exerts its own pressure as if all the other gases were not present. Henry's law states that the quantity of a gas that will dissolve in a liquid is proportional to the partial pressure of the gas and its solubility, given that the temperature remains constant. The partial pressure of a gas is the pressure exerted by that gas in a mixture of gases. It is symbolized by Px, where the subscript is the chemical formula of the gas. In internal and external respiration, O2 and CO2 diffuse from areas of higher partial pressure to areas of lower partial pressure. External respiration or pulmonary gas exchange is the exchange of gases between alveoli and pulmonary blood capillaries. It depends on partial pressure differences, a large surface area for gas exchange, a small diffusion distance across the respiratory membrane, and the rate of airflow into and out of the lungs. Internal respiration or systemic gas exchange is the exchange of gases between systemic blood capillaries and tissue cells. So this picture shows changes in partial pressure of oxygen and carbon dioxide in mmHg during external respiration. Pulmonary capillaries are there. Atmospheric air PO2 159 mmHg, PCO2 0.3 mmHg, and O2 exhaled, O2 inhaled. Alveolar air PO2 105 mmHg, PCO2 105 mmHg. Next, transport of oxygen and carbon dioxide. Oxygen transport. Oxygen does not dissolve easily in water, so only about 1.5% of inhaled oxygen is dissolved in blood plasma, which is mostly water. About 98.5% of blood oxygen is bound to hemoglobin in red blood cells. Each 100 ml of oxygenated blood contains the equivalent of 20 ml of gaseous oxygen. The heme portion of hemoglobin contains 4 atoms of iron, each capable of binding to a molecule of oxygen. Oxygen and hemoglobin bind in an easily reversible reaction to form oxyhemoglobin. So Hb plus O2, reduced Hb plus oxygen, binding of O2, dissociation of O2, reverse. HbO2, oxyhemoglobin is produced. The 98.5% of the oxygen that is bound to hemoglobin is trapped inside RBCs, so only the dissolved oxygen 1.5% can diffuse out of tissue capillaries into tissue cells. The most important factor that determines how much oxygen binds to hemoglobin is the PO2. The higher the PO2, the more oxygen combines with Hb. Then reduced hemoglobin is completely converted to oxyhemoglobin. The hemoglobin is said to be fully separated. When hemoglobin consists of a mixture of Hb and HbO2, it is partially saturated. The percent saturation of hemoglobin expresses the average saturation of hemoglobin with oxygen. For instance, if each hemoglobin molecule has bound 2 oxygen molecules, then the hemoglobin is 50% saturated because each Hb can bind a maximum of 4 oxygen. The relation between the percent saturation of hemoglobin is illustrated in the oxygen hemoglobin dissociation curve like this. PO2 in the x-axis, percentage saturation of hemoglobin in the y-axis, oxygenated blood skeletal, then oxygenated blood, oxygen hemoglobin dissociation curve. Look at this picture. Next, other factors affecting hemoglobin affinity for oxygen. Although PO2 is the most important factor that determines the percentage of oxygen saturation of hemoglobin, several other factors influence the tightness or affinity with which hemoglobin binds oxygen. In effect, these factors shift the entire curve either to the left, higher affinity, or to the right, lower affinity. The following three factors affect hemoglobin affinity for acidity, pH, and as acidity increases, pH decreases. The affinity of hemoglobin for oxygen decreases, and oxygen dissociates more rapidly from hemoglobin. The main acids produced by metabolically active tissues and lactic acid and carbonic acid. When pH decreases, the entire oxygen hemoglobin dissociation curve shifts right. At any right in the sense lower affinity. So at any given PO2, Hb is less saturated with oxygen, a change termed as Bohr effect. Partial pressure of carbon dioxide. Carbon dioxide also can bind to hemoglobin, and the effect is similar to that of H+. As PCO2 rises, hemoglobin releases oxygen more rapidly. PO2 and pH are related factors because low blood pH or acidity results from high PO2, PCO2. As carbon dioxide enters the blood, much of it is temporarily converted to carbonic acid. A reaction catalyzed by the enzyme in red blood cells called carbonic anhydrase CO2 plus H2O giving H2CO3, carbonic acid plus H plus plus HCO3 minus, hydrogen ion and bicarbonate. The carbonic acid thus formed in red blood cells dissociate into hydrogen ions and bicarbonate ions. As the H plus concentration increases, pH decreases. Thus, an increased PO2 produces a more acidic environment which helps in release of oxygen from hemoglobin. So, BPG, a substance found in red blood cells called 2,3-biphosphoglycerate or BPG, previously called diphosphoglycerate, decreases the affinity for hemoglobin for oxygen and thus helps unload oxygen from hemoglobin. When BPG combines with hemoglobin by binding to the terminal amino groups of the two beta globin chains, the hemoglobin binds oxygen less tightly at the heme group sites. The greater the level of BPG, the more oxygen is uploaded from the hemoglobin. Carbon monoxide poisoning. CO binds to the heme group of hemoglobin just as oxygen does, except that the binding of carbon monoxide to hemoglobin is over 200 times as strong as the binding of oxygen to hemoglobin. Elevated blood levels of CO causes carbon monoxide poisoning. Then carbon dioxide transport. Under normal resting conditions, each 100 ml of deoxygenated blood contains the equivalent of 53 ml of gaseous carbon dioxide, which is transported in the blood in three main forms. Dissolved carbon dioxide. The smallest percentage, about 77%, is dissolved in blood plasma. Then carbamino compound, a somewhat higher percentage, about 23%, combines the amino groups of amino acids and proteins in blood to form carbamino compounds. Because of the most prevalent protein in blood is hemoglobin, most of the carbon dioxide transported in this manner is bound to hemoglobin. The main carbon dioxide binding sites are the terminal amino acids in the 2 alpha and 2 beta globin chains. Hemoglobin that has found carbon dioxide is termed carbaminohemoglobin. So, somewhat higher percentage, about 23%, combines with the amino groups of amino acids and proteins in blood to form carb amino compounds. Because the most prevalent protein in blood is hemoglobin, most of the carbon dioxide transported in this manner is bound to hemoglobin. The main carbon dioxide binding sites are the terminal amino acids in the 2 alpha and 2 beta globin genes. Hemoglobin that has bound carbon dioxide is termed carbaminohemoglobin. Hb plus CO2 or carbon dioxide gives HbCO2, carbaminohemoglobin. Then bicarbonate ions, the greatest percentage of carbon dioxide, about 70%, is transported in blood plasma as bicarbonate ions. As carbon dioxide diffuses into systemic capillaries and enters red blood cells, it reacts with water in the presence of the enzyme carbonic anhydrase or Ca to form carbonic acid. It dissociates into H plus and HCO3 minus. So CO2, carbon dioxide plus H2O, water, creates carbonic acid H2CO3. Then H plus hydrogen ion, then HCO3 minus bicarbonate ion. Thus, as blood picks up carbon dioxide, bicarbonate accumulates inside RBCs. Some bicarbonate ions moves out into the blood plasma down its concentration gradient. In exchange, chloride ions move from plasma into the RBCs. The exchange of negative ions, which maintains the electrical balance between blood plasma and RBC cytosol, is known as the chloride shift. The exchange of negative ions, which maintains the electrical balance between blood plasma and RBC cytosol, is known as the chloride shift. The amount of carbon dioxide that can be transported in the blood is influenced by the percentage saturation of hemoglobin with oxygen. The lower the amount of hemoglobin, the higher the carbon dioxide carrying capacity of the blood, a relationship known as Haldane effect. The lower the amount of oxyhemoglobin, the higher the carbon dioxide carrying capacity of the blood. A relationship known as Haldane effect. Two characteristics of deoxyhemoglobin give rise to the Haldane effect. Deoxyhemoglobin binds to and thus transports more carbon dioxide than does HbO2. Deoxyhemoglobin binds to and thus transports more carbon dioxide than does HbO2. Deoxyhemoglobin also buffers more H plus than does HbO2, thereby removing H plus from solution and promoting conversion of carbon dioxide to carbonic acid via the reaction catalyzed by carbonic anhydrase.